Born on July 25, 1920 in London, Rosalind Franklin was one of the most accomplished yet underrated chemists of all time. During her time in England and France, Franklin used X-ray crystallography, which employed a high-resolution microscope that allowed her to see protein structure down to the atomic level, to study the structure of DNA. This research played a crucial role leading to the discovery of the molecular structure of DNA, what we now call the double helix.
During her graduate studies at King's College, Rosalind Franklin worked to improve the X-ray crystallography unit. Franklin was able to take two sets of high-resolution photos of two different kinds of crystallized DNA fibers varying in their levels of hydration. One of these photos, photo 51, created a clear picture of DNA's three-dimensional structure by displaying the way a beam of X-rays scattered off a pure fiber of DNA. By using DNA fibers with low water content and others with high water content, Franklin found that these two conditions produce different diffraction patterns. By improving her methods of collecting DNA X-ray diffraction images, Franklin obtained photo 51 from an X-ray crystallography experiment she conducted on May 6, 1952. First, she minimized how much the X-rays scattered off the air surrounding the crystal by pumping hydrogen gas around the crystal. Because hydrogen only has one electron, it does not scatter X-rays well. She pumped hydrogen gas through a salt solution to maintain the targeted hydration of the DNA fibers. Franklin tuned the salt concentration of the solution and the humidity surrounding the crystal to keep the DNA entirely in one form. After exposing the DNA fibers to X-rays for a total of 62 hours, Franklin conducted the resulting diffraction pattern and labeled it number 51. That became photo 51. This photo demonstrated the double helix structure of deoxyribonucleic acid, the molecule containing the genetic composition of all living organisms. Franklin's research is a perfect example of how basic science can lead to important discoveries. Franklin's work was really to improve X-ray crystallography and to explore basic questions about DNA, and yet her work led to numerous important applications.
2 years later, a colleague of Franklin released the famous Photo 51 and her unpublished data without her consent to fellow scientists James Watson and Francis Crick. Realizing that the photo and her data aligned well with their own data, Watson and Crick published a paper in which they demonstrated their DNA model, as they called it. For this work, Watson and Crick received the Nobel Prize in 1962. Franklin, at the age of 37, died of ovarian cancer due to extensive exposure to radiation in the lab, and her contribution to the DNA puzzle went unrecognized. Even in Watson and Crick's paper, her name was nothing but a quick mention. Discovery of the DNA structure has hugely motivated scientists to better understand the human genome and lay the framework for future research and medical innovations, especially in the field of pathology and virology. For instance, the rapidly evolving HIV virus has been bugging scientists for a long time. But with knowledge on the DNA structure, scientists are now able to track the evolution of the virus within individual patients and therefore tailor treatment accordingly. It also helps us trace the origins of HIV pandemics and work out the transmission history within and across countries. In addition, using DNA sequencing, we are now able to identify the genetic information carried on a specific segment of DNA. And this is hugely important, since it lets us to not only spot genetic diseases almost immediately, but also identify their genetic causes. For instance, some types of cancer, mental disorders, and Alzheimer's disease have been found to be associated with certain DNA segments. So, what have we learned? Franklin's research started as a simple quest to answer two questions. First, what lies beneath the surface of DNA? And then, how does DNA form in the first place? Her curiosity led her to try out different methods of experiment, not for profit or reputations, but for a genuine desire for scientific knowledge. It is that curiosity and creativity that fueled the subsequent discovery of double helix, the development of DNA sequencing technology, and many other modern technologies. With or without a Nobel Prize, Franklin's passion and contribution to basic science should not be forgotten.